So if you run through the end of the practical, then you know how to open an fMRI file, right? How to look at it with Python, how to do initial analysis, i.e. take the average across time for some time period, or take the standard deviation, do some things like that. But a standard fMRI analysis, the ones like we need to teach you because are basic, and the ones that you will see happening in the YouTube snippets that we also put online, they're a bit more involved. So the second and the third practical will go into how an analysis like this works. But what I'm going to do today is really go through the basics of why we need to do the type of analysis that we end up needing to do. So the idea is that by the end of next week, you'll have a basic idea of how to interpret and read and understand most of the literature that was published until around 2012. So then the remainder of the course after next week will be to show you how fMRI has evolved since then. So you'll notice is that there's a lot of focus in the YouTube clips that you can peruse on, that we link to on Canvas. There's a lot of focus on this localization of activation. There are already some Discord questions also, so we'll also go into what the limitations of that are. What we'll be talking about today is really that sort of basic idea behind why we need to do this specific type of. So the first thing we need to talk about is this hemodynamic response function that we've already talked about a bit, right? We have a clear understanding now of how there's a difference between this thing that we're interested in, namely neural firing, or the thing that we think corresponds to our experience, right? Neural firing, neurons communicating with one another, creating the mind, if you will. And then the question is, how do we use the fMRI machine to get some sample of that? Well, this is an inherently sort of ambiguous thing, right? There's a lot of distance between the bold response that we measure and the neural firing that we are interested in, right? So this is one of those caveats. And so what I'll be talking about today is how we can still use the principles of the shape of the bold response and things like that to infer something about neural function. So what's the type of analysis that we do? This thing worked like 5 minutes ago. Okay, so as you apparently as you saw before, right? This is that movie that I showed you before. This is a brain recording, right? You know the different slices. And what you're seeing at the bottom is the time course of a selected voxel somewhere in the brainstem. I think this is that respiratory signal that I talked about before, right? So whenever the subject breathed in, it moved the bottom of the brain a bit, and whenever they breathed out, it also moved the bottom of the brain a bit. So when you pick a voxel in a region like that, you see a lot of this back and forth. We think that's related to breathing. But of course, as we clicked through, right, we saw different sort of signatures of activation. And then what we want to be doing is conduct an analysis that allows us to start to find things that relate to our experiment. Here's our breathing again. I think at some point I'm going to click where there isn't necessarily breathing, but a more functional response. So I'll be waiting for that. And there you actually see these activations go up and down in a very systematic way. And we as experimenters know what happened when in the experiment. And so that means that we can now start to relate the brain activations that we're measuring, such as these. These are the signal that we're interested in. We knew that we're presenting something here and here and here and here, right? We want to be able to say, this happened in the experiment. Now I expect this sort of signal to transpire in the brain. So that is the signature of the response that I'm going to be looking for. So the end result of an analysis like this, where you want to see, for example, if I showed a face we had this activation. If I showed a house, we got this activation, and I want to say which voxels respond to a to faces when I present them. So I'm just going to use this example throughout the lecture of presenting faces versus presenting houses, because there are specific regions in the brain that respond to when I show you a face or when I show you a house. They're very nicely segmented. So I'll be using that as a sort of model system. The idea is, how do we get from this raw data, right, and this bucket of noise, to analyze it and then get to an output like this, which is a statistical map where we're coloring something orange, red, yellow, if it responds to houses, and bluish and towards lighter blue if it responds to pictures of oh geez, I made an error here. I think yeah, so this should be faces. On the right, blue is faces. I'm sorry. So what does this look like? Well, it turns out that if you go into the literature, we actually have a whole network of face selective regions in the visual brain. So this is responses to pictures of houses and faces, right? We have whole networks for houses, and we have whole networks of faces, which makes sense, right? Faces are important because I need to know how bored you are, right? So I have to be very good at, seeing the little hallmark signs of boredom in your faces, and it requires a lot of processing, which is a beautiful scientific question in and of itself, of course. Now, the houses are also very important because you need to walk into the right house as you, come home from a night of partying, for example, right? So they're also very, very important classes of stimuli. And so if I just to highlight how such a outcome, what such an outcome looks like, right? If you look at what are my responses to houses, what are my responses to faces, you really see these nicely sort of interdigitated patches in the back of the head where we know the visual system is. And so this is the sort of result that you get from a standard analysis. Yeah, so you get, when you presented houses, when you presented faces, for example, in a movie. And then when house happens in the movie, we say, now I expect a response, right? Whenever a face happens in the movie, we also say, now I expect a response. And so we can compare these expected responses to that bucket of noise. And this is the type of outcome that we get. Yeah, so more or less empty brain, and these patches of what in the sort of popular science literature people refer to as activations, right? But they're not activations, we're looking at statistics, right? So this is the end result of a pipeline of analysis steps. And so what we're not looking at is a neural response, because we were looking at a bold response. There is an indirectness there that I really want you to deeply appreciate. And then whenever you see patches in the brain like this, so this is really that localization approach to fMRI. If you see someone saying, oh, this is the love region of the brain, you're not looking at activations in that region. You're looking at the end result of a statistical analysis where it's not like a bold response or percent signal change or neural activation. You're looking at a statistic, most likely some form of t-statistic. Yeah. So those of you that are statistics aficionados will get to your hobby of choice in later lectures primarily. Yeah. So this analysis that I'm going to try to explain today is all about creating an expectation of what a signal should look like, and then comparing it to the actual data. Yeah? So there's two major parts here. The first is that we need to construct that expected response. And then the second is, how do we find the relevant strengths of different types of responses that we can model? Yeah. So I'll get to those two different stages along the way. The first then is to know what type of time course of signals to expect. Yeah. That's the first. And of course, the most important aspect that we then need to take into account, apart from when I showed that face and when I showed that house, is the shape of the hemodynamic response function, this thing that you've already seen pass by, right? We have this slow response that we know reflects this weird response of the vasculature that has to do with the deoxygenation of the blood, right? So it's this blood oxygenation dependent signal. And this is just to highlight the different phases of that response, even in the tissue. I talked about how earlier on, I talked about how you have this arterial side that supplies the blood, and you have a venous side that sort of draws away the expended blood, if you will. And this is a very beautiful sort of cascade of physiological responses. So you get widening of the capillary veins, so the total hemoglobin increases. You get arterioles that dilate, so they start allowing more blood to pass in. So you get this whole sort of flow response in terms of arteries and veins. At the same time, oxygen is extracted from the blood locally because neurons are consuming it, right? And this is what gives us this incredibly complicated physiological cascade that we just have one reflection, right? We just have this bold response that we measure with fMRI that goes a bit like this. So what that bold response is most influenced by, what it really like deeply down reflects, is this decrease in oxygenation over time that you see happening there. Yeah. But it's important to realize that this is a very, very complex response. Even though what we're picking up is deoxygenation of hemoglobin, there is all sorts of flow components, there's all sorts of volume components in that response that we can sort of zoom into with different types of measurement. That's what we talked about last time. I hope that sort of rings a bell for some of you. What that means is, of course, like I also said last week, and I hope that Nikos will have some time to talk about it next week also in the MR physics lecture, is that we can use the scanner in different ways, right? The standard way of doing fMRI is to focus on this specific characteristic of the bold response, right? So this decrease in hemoglobin, but you can also try to measure things like cerebral blood volume that isn't sensitive to blood oxygenation, for example, right? And so there's many different ways in which you can use the scanner, like you saw in different anatomical images. Same goes for functional MRI in principle. What we do mostly is this bold response focused on this hemoglobin, right? Originating from this change in hemoglobin oxygenation. And so that's what we'll talk about mostly for the remainder of the course, right? So when someone says fMRI, they will generally mean bold dependent fMRI, but there are others. So what is this shape of the bold response? Yeah, what does this mean? What does it all mean? The bold response looks like this. And let me just go through these three important phases of the bold response. We have this first initial dip that you see happening here, and it is elusive. No one ever says that they've focused an analysis specifically on this. No one ever reports analyses that are based on this initial dip. Yeah? So in terms of how we're looking at, we think that this does exist, and if you do a very specific measurement of it, you will find it, but a general analysis will not take it into account. So if you look at the sequence of events in terms of that T2 star development of the signal over time, you expect this first initial dip, and that's that decrease of the oxygen, or so that the initial oxygen being used, and the vasculature with its fast response responding to that. Yeah? Then you get this really, really big main bold response, and that is the aspect of this response that most analyses are most focused on. Yeah. So even though you see a bit of an undershoot there, which does exist, and that really reflects sort of the compensation for this oversupply of new blood and things like that, we have this negative part there. Our model of the hemodynamic response function does incorporate it. The main bulk of what we try to explain is all based on that main bold response that peaks somewhere between 3 and 6 seconds. Again, this is the reason why we cannot do very fast experiments. Yeah, if you want to do like sub-second precision experiments, very, very difficult with fMRI. So you can do it, but you need to like really know your stuff, sort of thing. Okay. Now, what is this bold response, right? It's nice that we say this, but okay, but what was this response caused by, right? And one of the major aspects of what I'll be talking about in this first part of the lecture is really that this response, we hypothesize it to be the result of an infinitesimally small activation of neurons happening at time 0 here. So if you think about the neural response that happened, well, our brains are always sort of active, right? This is coming from the imaginary situation where you have 0 activation in terms of neurons, and then all of a sudden, for a very brief period of time, you have maximal activation, and then it's gone again. Yeah. And so then the idea is, what is the shape of the response in terms of the bold signal that we measure over time? Well, that's this. Yeah. So it's a sort of imaginary thing, and we call that thing an impulse response function. I'll get into how we, why we need to think about this in this way later on during the lecture. So, yeah.
I was just wondering if there's a way to tease apart the time scales between the activation of the neuron and the bold response.
Mm-hmm.
Is there a way to do that?
Well, it's a funny you should ask. So there are, so it's very hard to dissociate, like, because you need a view on the neurons to be able to do this sort of thing, right? But it turns out that about a month ago, 2 months ago, maybe 3, I don't, some months ago, very brief, like not that long ago, there was a beautiful science paper, like, so Science, right? One of the highest impact journals. If something appears in Science, it's going to have a lot of impact on the field. And there was a science paper that showed that in rodents, at least, they could use the MRI scanner to what they pretend, or not pretend, but what they think is to look at neural activations. So they do away with this whole bold response that lasts up to 30 seconds. They say, if we use the scanner differently, then, so this is all like in the millisecond time scale. So they have an experiment where they use the scanner like really, really fast, and they're able to look at a response that isn't 30 seconds, but 30 milliseconds. And they actually showed that you get a similar type of response. It's shaped differently. And then they control for, like, maybe this would be blood related also, right? And they try to control for that. And so I haven't really been able to dive really deeply into it. I'm sort of involved in trying to replicate this in humans because this is only done in rodents. So in principle, you can have, this is one of those things, right? We don't know. In principle, if you're a smart sort of MR person, maybe you can find something in the MR signals that gives you evidence of like precise neural activations. And this might be one of them. So people are really diving into this, right? If you go to a conference now of fMRI, like the technical side of things, people are really going all in to try to see if they can replicate this in humans, because of course, then like half of this course would be completely trivial. I'm sorry, but then I would just be teaching you totally, yeah, you wouldn't need to know all this stuff anymore, because we could actually image neural activations, right? Of course, there would be all sorts of caveats, but yeah, so there are breakthroughs reported, and some people have sort of claimed that this is the case for many years, but no one believes them. It's also that sort of field. But yeah, now there seems to be a way where we can measure neural activation, like at the very, very short time scale. But yeah, for now, that is all very sort of dreamy, future oriented. No one can do it in humans sort of level. And so we are blind. We can only look through the blood-soaked lens of the bold response and thereby get an idea of what happens in the neurons. Yeah.
So do we then not know how long or short the burst is?
Yeah, so this is an imaginary one, right? So what you can do is say, well, for all intents and purposes, I'm just going to show you a flickering something for half a second, right? And then I'm going to say, like, if I expect a response of like 30 seconds, then half a second is, for all intents and purposes, like pragmatically, is infinitesimally brief. Yeah. And so that's what you do. And then you can sort of estimate the bold response shape. Yeah, I'll get into estimating that shape in a bit later, but that would be the approach. Yeah, so This is also like a bit of a physical, like physics thing, right? So there's this joke of a physicist getting asked to optimize milk production. Like a farmer comes to a physicist, and then the physicist does also these calculations, and he goes to the farmer and says, okay, I have an idea of what you should do to optimize milk consumption. First thing you do is you assume a spherical cow, right? And so that's what you do, right? And then you start running with it, and you see how far you get. And this is similar, right? You just assume that something is infinitesimally small, and then you see how far it gets you. And then it turns out that in many cases, you wouldn't have to worry about all the details so much. Yeah. The spherical cow won't be exam material. Okay, so we're looking at this bold response, right? So there was a whole segue here into like neural activations. That's all very recent, also not part of the course, right? That's just a segue. Okay, so just a few words about the shape of this bold signal. It's slow, right? So if you want to do a fast experiment, do EEG or MEG. And so people that do EEG and MEG, they will say our measurement device, our measurement modality, EEG, has millisecond time decision. And they go like, yeah, right? Of course, then we have all these problems with there's a different frequency bands and not knowing exactly where things are in the brain, right? So every measurement modality has its problems. And the problem with bold fMRI is that it has very bad temporal resolution. Yeah, it then excels in spatial resolution. Right now, what people do is they always show this bold response, right? And here's another caveat. I can show this bold response, and it will look more or less similar for all of you, right? But it varies. The shape varies tremendously across. Well, I think I have a list here across different people, right? So that depending on the subject that you record from, the bold response will differ. According to age, as you get older, your bold response becomes less strong. I wish it wasn't the case, but your bold response gets lower and lower in terms of its maximal amplitude here. So you get noisier measurements in the end. It will also like probably change shape according to age. We're actually doing an experiment right now to look at this, how this changes over age. We also know that the bold response shape depends on where you're looking in the brain. So we have been able to estimate the shape of the bold response, and I'll show you examples of how we do that, how we perform an analysis like that. But we've been able to estimate the shape of the bold response for regions like, for example, M1, so motor cortex, like that first motor region, because if you ask people to really quickly like clench their fist for about half a second or a second, and then do nothing for 20 seconds, of course you would get something like this, yeah? Or if I show you a flickering pattern really, really quickly and then stop, right? I would get a response like this. But if you're looking at, for example, like hippocampus, like a memory-related region, I have no way of presenting a stimulus that I know will be zeros for 20 seconds, then one, like maximal for like a half a second, and then nothing, right? How can I do that? I have no idea. Right? So in many regions of the brain that are deeper and that are responsible for more complex cognition, we only have a very vague idea of what the shape of the bold response looks like there, because we just don't have the experimental design or possibility to estimate that shape, to find it. And it's like a pragmatic thing. We just cannot, because we cannot say for certain that this thing will be quiet for half a minute and then maximally activated for about a half a second, right? You can only get that sort of situation if you have full control over what's being done in that region, which leaves us with V1 for visual stimuli that you have full control over, M1 for like motor tapping tasks, A1 for bursts of sound, that sort of thing. Yeah. And so what do? Well, we just sort of pretend that we know the bold response there, and we assume that it's the same as it was in the regions that we did have control over, right? So caveat number, I don't know what. I'm going to make a list of caveats at some point. Yeah, you can see how that can be a problem if you want to look at responses deeper in the brain, for example. So we don't know the shape of this bold response for some brain regions. Now, we also know that in many cases, your cognitive state will change the shape of the bold response. I'm now saying things like attention, but for example, your the link between this neural firing and the bulge response changes as a result of dopaminergic transmission, right? So we talked about that in one of the previous lectures. So the shape of the bulge response also likely changes depending on whether a stimulus is rewarding or not, things like that. Yeah. And so in all of these cases, we generally tend to assume that the bulge response has a stereotypical shape that does not change according to all of these things. And there are more. Yeah.
What happens if you show like an image, half a second, then 10 seconds later, show the image again? The bulk response is 30 seconds. So today overlap.
Yeah, they overlap. And and I'll get to this so it's a good question. I'll get to this in a few slides. So one of the central tenets of this main analysis, the general linear model analysis of fMRI data, is that these can be considered as independent events that just sum in terms of their response. Yeah, so you will get if you show something here, right? You will have a signal that starts here, and then you will get a dip, and then a bulge response there, and then it will continue like so. That's the general idea. But I'll get into that in a bit more depth in a minute.
The statement you were mentioning about how you could have a baseline response and then the sudden showing of an image is going to show the relative distance. Can there not be something like that for memory or something? A trigger to think of something like an automatic.
Yeah, that would be really cool. So maybe I'm being a bit dismissive implicitly about memory research. I don't know, and I apologize for anyone that I'm insulting. I think it's just a bit harder, right? You don't have much control about what people are remembering at any point in time, right? So it's also a matter of can you clearly state that you had 0 memory-related activations for half a minute? And I think that statement is hard to strongly defend. You might come up with something that mimics a situation like this, and then you would have a mapper for the shape of the hemodynamic response in things like the hippocampus. Yeah. It's also that if you don't already know what the function of the area is, you don't even know what that stimulus at time 0 should be, right? So it's also a sort of a chicken and egg problem. Yeah. Good point. Yeah. So we know that if we pretend that there is a standard bulge response, and we do pretend in most of our analyses, right? Caveat X. We know that we're missing things, right? And so at the end of the lecture, I hope to be able to show you a bunch of methods that we do have to be more flexible in this regard, to not have this assumption get in the way so much. But that's for later. I do want to, again, because there's this push in the field to go to laminar fMRI, right? And people talk about that a lot. I want to highlight that. There is also a difference between the shape of the HRF in different depths of the cortical tissue, right? So if your voxels become smaller and smaller, you can get to lower and higher regions inside the surface of the gray matter, right? So you get like closer to the white matter and further away from the white matter. And we know that the shape of the bulge response also depends on where you are sampling in that cortical depth, right? So you get much stronger bulge responses if you're closer to the outside of the gray matter, and much lower responses if you go closer to the white matter. This is because of those draining veins, right? So at the top of the gray matter, that's how the vasculature works, right? The larger vessels are at the top, so they drain. That's where the blood drains towards, right? This deoxygenated blood. So that's why you get a stronger signal. Now you can imagine that if the blood actually gets used here and then flows up, you wouldn't just get a lot of signal there, but anyone have an idea? There's another thing that might happen if you have to, you're seeing draining veins at the top of the gray matter. Yeah, so you do get accumulated noise, but there's another factor that I think, so that's a good point. You do get higher amplitude noise also, but you get higher amplitude signal, and there's a bit of a trade-off there. That's complicated. Good point, though. There's another one that I was sort of aiming for. Yeah. Yes. So it's later, right? So what you actually have is that the shape of the bulge response really reflects when the blood in terms of its deoxygenation, arrives where you're measuring it, right? So, and it can be the case that it already gets depleted here, but then takes about a second or two to arrive at the top. Yeah. And so you're really looking at this sort of, flow. You're looking at this slow sort of waterfall-like oxygenation thing happening in the tissue. And that's what you see here, right? So if you are looking in a large vessel, you actually see that the peak of the Bold response can be delayed up to 2 seconds relative to when you're looking at the small vessels that are really close to where the neurons are firing in terms of this hemodynamic response. Yeah. So, yeah, we have this blood drain going up there. Yeah. So, again, caveats galore, right? It's we assume generally the shape of a Bold response, and most analyses just say we have a fixed shape across everything and everyone in all brain regions. But we need to always keep in the backs of our minds, this is an assumption. Yeah. I keep doing this with the caveats. I really like it. It's I'm gonna keep figuring out things that I should call caveats, because it's good to make that explicit. I'll make a list and make sure that I have a caveat list before we get to the exam. Yeah. There's a lot to, understanding a field also means that you have ways of deconstructing the things that people do in a field. And I think the caveat list might be a good vehicle for that. Okay, so we had this question just now. Any question about what we just talked about, the shape of the hemodynamic response function, how it's complicated? Great. Now let's say we feel comfortable for now, assuming a fixed shape of a Bold response. Let's say, okay, we'll just have a fixed shape. This is spherical cowl. We'll just keep working with it for now, right? And we'll take it from there. And how do we then use this shape of the Bold response in a subsequent analysis, right? Then we want to start using this spherical cowl of a fixed Bold response shape and see how we can get to milk production. Yeah, I'll stop with the metaphor. And so we need to take into account a bunch of assumptions that then allow us to start working with this. So let me take you to one of the things that you've already seen, the sort of idea of a basic analysis strategy. I think you've seen this in a previous lecture, the data that I'm going to show you. Right, so the idea is that stuff happens in an experiment, your neurons fire, and then you get Bold responses that also change over time, right? So you get these beautiful Bold responses, and this is that first human experiment of fMRI, right, with a flickering stimulus on the visual cortex, and we saw this is just an arbitrary signal intensity, right? Like I already said, the number that you get out of the scanner is in principle meaningless, right? We have these numbers, and we see that when the stimulus is off, it's low, and you get this beautiful Bold response. It goes up, right? And then the stimulus is turned off, and it goes down, on, it goes up, right? Beautiful over the course of 4 minutes, right? We have a minute-long flickering checkerboard is what people use in this type of context. It's headache material, but it gives you a strong Bold response. Yeah. And you can see that if you do a simple analysis like that, right, you have a slow experimental design, and you have a very strong visual stimulus. In this case, if you're looking at visual cortex, it's very easy to analyze. You can immediately come up with easy ways to analyze. Because very simply, what you can just do is take these blocks, for example, and because one is on, one is off, right? You can just take them and then put them together, right? And I'll say, oh, I have 2 samples now, right? Now I can do a bunch of stuff and start. And then here comes the core of this. We can start averaging things, right? So we can average everything that happened when the stimulus was on and say that this is the Bold response when the stimulus was off. Bold, on. We can do the same for when the stimulus was off, so we get the value for when the stimulus is not on, and you can subtract the 2, and that's our Bold response strength, right? We could even do this across multiple runs, for example, and then do statistics. For example, we could just t-test this difference. That's per voxel. You get a t value and a p value per voxel, and you can get these blobs that we were talking about. Beautiful. This is the basics. You're done. Right? And this does hold. This is valid. This works. Yeah. But you can definitely see what a problem here might be. Anyone? Oh yeah, so this is averaged across voxels, which is why it looks so nice. That's a problem. This is a specific type of analysis that we call a region of interest analysis, and we'll go into that in the practicals also, why that would be different from just doing analysis on every voxel. So good point. I'm talking about, though, I was targeting for another one that you can see from the time scale here. Yeah. Yeah. So you have a very slow experiment, right? And you already see that here you have this little overlap between the off response and the on response, right? So what you're doing is you're epoching the data, right? And what that means is that because of the slowness of the bold response, and I'll get into this in detail in a bit, because of the slowness of the bold response, it's very hard to make sure that you have an epoch that doesn't have any contamination from a previous event, right? And what this means is we are forced to do extremely slow experiments, right? So if you want to do averaging analyses, you are stuck with, cognitive processes that you can actually do for a minute or so, right? And then not do them for a minute or so, right? This is incredibly coarse, right? You're losing any time, right? And we know that the bold response is slow. We know that if you're interested in fast cognition, you should use EEG. But this is a bit bad, right? Yeah. Nonetheless, though, if you, again, faces, yeah, sorry. Nonetheless, you can produce things like this just from this averaging, right? If I showed faces for a minute and houses for a minute, right? I could still get responses like this. It just means that I would be putting people in the scanner for an hour instead of maybe shorter. And so I would lose the ability to do interesting experiments. Yeah. Now let me get into this problem with averaging, because it's definitely a problem, right? It forces us to only do extremely slow things, and we want to go faster. Yeah? So let me show you an example, right? So let's have an example situation where we are looking at a voxel, single voxel. And so all of the analyses, most of the analyses that I'll be showing you today are all on the single voxel level, right? We're only interested in a single time course, just to show you the principles of why we're doing analysis the way that we are. And then later on, we'll go to how you then do this across the entire brain. That's also what the practicals will then show you how to do. So we're going to simulate this. It's also a very important principle. If you can simulate something and then understand the results, it means that you have a big, like a deeper grasp of what happens than just doing exploration on something that you don't know, right? If you can create it, then you understand it, sort of thing. So we're going to simulate things. We're going to simulate the time course of responses to two different types of events, cues and stimuli. These are just made up. They could be sounds and vision. They could be houses and faces, no matter. And so what we're also going to assume is that the stimulus response has a positive bold response, like so, with a specific shapes. And we're also going to assume that the cue-related response has a negative bold response. This can also happen. Be open to the possibility that, some responses might be negative. So if we have these two types of responses, two infinitesimally brief events happening throughout our experiment, what we're going to do is just see what happens when we use this assumption that we can just add them all in time, right? So if they happen here in time, I'll get this response. Another response happens, like for example here. Another response happens just a bit later. I can just take their responses and then average them together. So what you see here is that you get this beautifully weird response. We've also added a bit of noise to make it look realistic, like actual data, right? So who knows whether this is actual data? I do as a simulation, but so you get this beautiful wavy pattern in the bold response. Yeah, and this is what we want to find out, right? So if we have a voxel that we're recording this from, how can we analyze this? Yeah. So this is a very simple experiment. These two different types of events are interspersed and they happen at different moments in time, right? So here the cue is right before the stimulus. Here it's a couple of seconds removed, right? There's this delay that's variable. Yeah. Now this is cognitively a lot more interesting, right? If you could analyze a time course like this, that's very complicated. If you could analyze it effectively, then you would get a lot of inference power, right? As a scientist, you could do much more interesting experiments, and that's really the goal here. Yeah? Right, but so first, let's analyze this in the way that we just highlighted that people started out with, right? When we just had the scanner, we just had these bulb responses, 60 seconds, epoch the response, right? So that is to just take the data after a given event, take those data and then just put them in somewhere else, and then take for the same type of event, take that same 20 seconds after the event, and then just average all of those event time courses, yeah, those 20-second event time courses. So what that looks like for example, the Q response, the red one here, I just took 20 seconds after this event, 20 seconds after this event, 20 seconds after that event, and that's what you see. You get this response, and the dashed lines are each of those repetitions, and the solid lines are the average, right? I can do the same for the stimulus locked response. You see that the response here was a bit bigger than that one, so you get a bit of a clearer thing. But you see the problem here, right? Anybody want to muse about what's happening here? Yeah.
They get a total average and not just an average of the Q.
Exactly. This is sloppy, right? You are throwing out, or actually you're calculating in the baby with the bathwater sort of thing, right? What you know now is that in those 20 seconds after a cue, in all of these cases, you will have a stimulus, right? And what that means is if you take these 20 seconds after that cue, what you're going to do is you're going to nicely look at a cue response, but you're going to have these random influences of the stimulus response that are also in there, and you've just blinded yourself to that effect, right? And similarly, the same holds for the stimulus response. You see that it goes down really quickly. There's that, right? Goes down really quickly. That's not part of that response. Well, clearly, that is just that ongoing cue-related response that always happened right before, right? The really strange or really finicky thing here is that because of all these random delays, there's no way of reconstructing this, right? If they always happened at a fixed delay, maybe you could like do some inference, but this is always random, and the averaging has thrown out a lot of information there. Yeah. And so we need to come up with something else. Yeah. And now we're having a break. So for 10 minutes, you're going to be stuck with this problem. Yeah. Let's be back here at 55. And I will again go through the ingredients of what the problem is here and also a solution. Isn't that great? Yeah, go ahead. Yeah. Here, design of 3. So there's 3 holes. 3 students. That comes out brilliant. Does it work? They're diagonal. Oh yeah, I think you need to rotate 19. Yeah. Oh no, they are. Strange. They're different. The other way around. Yeah, this is working, right? It's great.
Yeah, for example, my mom. Like,
Large.
nice.
Yeah.
Not day. Yeah, go ahead, provided that you do so they, but you haven't both them now, and they have to. Say, yeah. That's fine. Yeah, I know. Now I, yeah, you have to go back to the non. And you have to basically for you know, like reaching countries on there, maybe they almost the other. And now it's really impossible to know. Well.
Second.
Yes, yeah, Mark, you know,
Maybe just.
even if they don't speak, yeah. I'm trying to fight fiction.
They always say hi. 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 Give me one. I have to find out what it's gonna be.
Yeah, I will announce this today. I think that it will be Friday. You know, so it comes down to the Germany, I mean, and so if you ever went down in there, that's not a big. Yeah, I know.
No, actually.
You know, I don't care. So. No, I think that will be fine, but you can do whatever you want. I don't want to be, but they want First of all, I don't want any group like garbage. Yeah, because now we can go like this by a white. And they don't want anything like garbage. Yeah, but just, they can go to the store. I think it's like, about, we are happy. And I'm going to.
Hi.
Ask you a question about
Yeah.
the first slide.
Uh-huh.
Why the service is later? I don't get it.
Oh, yeah. So if you look at the right of the slide, right? So the surface is, so you have a white matter surface and then the surface of the brain, right? So this is not, so in this figure, it's not really well named. So the surface that we're talking about is this surface, right? So
No, this service is.
at the top of the brain,
is
yeah.
a top-
And so all of the brain, all of the blood, when it gets used, it sort of gets pushed upward, right? And that's why they say surface voxel. So even if, so they have I think they have three lines here, right? So
yeah.
there's one is deep gray matter, there's surface gray matter, and there's a large vessel, right? Those three.
Yeah.
And what this means is that the deep gray matter is here, right? The surface is here, and then the vein is actually here. So
Yeah.
those are the three graphs that you're seeing. Is that clear?
Yeah, but why is it well delayed?
Why it will delay? Oh, so it's like when I open the faucet here, it takes a while for the water to flow all the way there, right? So it's like yeah. So one of the instead of like water in a sink, right? It will flow down, right? Here things flow up, right? So when you have neural activity at the bottom, right? It will open a faucet, right? It will open a flow, right? And then it flows down, or it flows up slowly, yeah? And so there will be a delay, right? You have activation and a bolt response in the deep layers,
Okay.
and then it will flow up, takes about a second, and then it will be at the top where there's also more pooling and you get a stronger signal,
Mm.
right? So it takes about a second or two to get from the lower parts to the top parts. That's how that works. Is that, yeah? So it's as if I open the tap, right? The water starts flowing, right? I see that it starts, if I have a measurement of flow and I measure it in the tap, it's immediate, right? If I measure it in the sink, it's about a second later, same thing.
Okay.
So really think of this as, it's a bunch of pipes, right? Pipes that are widening and there's flow in them.
So this figure is means when the large brain has this figure, it actually means the big brain has activation.
So yeah, so if so here the neural response was the
Yeah.
same, right?
Yeah.
It's just that where you measure it, yeah. And if you measure it in a vein,
Yeah.
you will see it stronger and you will see it later. Yeah, it's just because it takes a while for the blood to flow there. Yeah.
Okay. So we also have this question about this part, we don't really get it. What do you mean the defense?
Oh, yeah. So I think here I'm just posing the problem, right? I timed the break a bit badly.
Yeah.
This is just the problem, right? The solution will come later. So I will outline
We
the
don't really get the point of this.
of the slide. Yeah, so if I have a response, so my next slide is actually going to go into this. So wait for the next slide and we'll talk about it a bit more after I show you the next slide. It's a surprise.
Okay.
Yeah.
Thank you.
Yay!
Like the dog,
Yeah, that'll be €5.
Yeah, I'll pay you in gratitude.
Okay. Let's continue. I understand that my timing of the break is a bit off insofar that the problem with averaging, I actually have a sort of summary depiction of that, which you can see on the screen now. And this, I think, makes the problem of this epoching analysis much clearer. Right. So again, if you are doing EEG analyses, right, where what you measure is more or less instantaneous at the millisecond level, and you are only interested in what happens like 600 milliseconds after you show something, then you can epoch things, right? Because your next trial will only be in a couple of seconds. There's no problem with the contamination of the different trials responses with one another. So an epoch analysis is done in things like EEG, no contamination over time. But because of our slow bold response, we do have a problem with contamination over time, and we need to find a way to fix it. Right. So here's an example of what this looks like. The intermixing of these responses, it's a problem. Like the question was before, like, do you sum up these responses? Yes, you do. And it's a problem if you want to do epoch analysis, but we are going to be also using this fact later on in the analysis. So we end up with a signal that we measure that goes a bit like this, right? I've taken out the noise so that you can actually see this. And if I'm doing an epoch analysis, I'm going to take these different epochs, right? And what you immediately see here is that for these two epochs, I'm sort of calculating the same data twice, right? For these, I'm taking the average signal, and it's going to be part of this green epoch, but it's also going to be part of that orange epoch that just staggered a bit in time. So that's counting double. Why do these data get counted double, right? They're not worth double. No reason to think that they should be counted double. It's just an artifact of what I'm doing. Similarly, it gets worse. You can see where it gets worse in this analysis, right? In this scenario, it gets worse there. Data are counted 4 times, right? No inherent reason why signals on these time points should be so important. But if I epoch them like so, I am counting them 4 times, and that gives them overly strong weights in the end result. Yeah. So the point here is, and I hope this clarifies things a bit, it's to unambiguously say this is the response to this event or this type of event, it becomes impossible, right? Because you're mixing everything together in quasi-random ways. So a huge problem. Yeah. I want to go back to the assumptions that we used to create this this simulation, and that will make things clearer, right? And it also gives us a handle on why we can start to solve this huge problem. So let's go into the assumptions that we use to create the simulation in the first place, right? We talked about this infinitesimally small things and things like that. We're using a bunch of assumptions here that are focused on the question that we had from the back just now, right? So we assumed this fixed shape for every response. And that's something that we're going to keep doing. So we're not going to think that the bold response might change from the second to the third event, for example, of the same category, right? That would wreak havoc. We don't. We always assume that the shape of the hemodynamic response function remains the same over time. And I'll have a word for that later on. We also assumed that when we have multiple events lined up closely in time and their responses overlap, that we don't have to do anything very complicated. We can just sum them all up. Yeah. So those are the two assumptions. We can use a fixed shape all the way. And whenever we see this type of thing happening close together, we can just sum the responses. And that's what gives us these curves, the black one here. Now, there's a technical term for this, and it's like a first year physics type thing that some of you may be aware of or familiar with. But these two assumptions, they relate to the definition of a linear time invariant system. And you've seen this term, of course, also in the paper that you've been reading. And what this means is that signals scale linearly in amplitude. That just means that we can add them up. Yeah. Now, we can add them up in multiple ways that I just want to highlight specifically and separately. So the first way that we can add them up is by saying, oh, if a single infinitesimally brief event, right, happens at time 0, we get this Bode response. Yeah, so it's some Bode response, slow. And you see that it's not that strong. The idea then is if I can figure out a stimulus that's twice as strong or 3 times as strong, or 4 times as strong, yeah, I will also, and that's what you see here on the left, I will also have a Bode response that is 2 times as strong, 3 times as strong, 4 times as strong, right? So that they, if I measure a 10-fold strong stimulus, I will get a 10-fold strong Bode response. Yeah, it's linear addition. Yeah, that's what this means. Yeah.
What is this assumption based on? Or is it based on like previous experiments? Or-
Yeah, so this is so it's to be clear, right? It works the other way. It's an assumption, right? And we use this assumption as a sort of foundational assumption for our analyses. We need it. If we don't assume this, all analyses will be like super hard math. Like if we don't. So we're just going to use this. Now, of course, we can do experiments where we validate that this is the case, right? So like I showed you in that monkey, EFIS, and Bode response study, right? What we can do is we can vary the contrast of a visual stimulus, for example, right? Or the rate of finger tapping. We can, those types of things, they will make a stimulus X times stronger, right? In a sort of objectified way. And this type of thing we've done for contrast in vision. And you see that there is a large range of contrasts where this assumption of linearity holds. But there is a problem also. That is, if you manage to create a stimulus that is 500,000 times stronger, right? And you are already getting 1% signal change, it means that like your brain explodes when you respond to that stronger stimulus, right? And so there is always going to be a nonlinearity insofar that you cannot keep multiplying things without heads exploding. So there is so this is the one of the caveats for the use of this LTI assumption is that there is always a limited range in which you can keep assuming this. Yeah? And you will transgress into regions where things become nonlinear. And there are, like I said, very complex math ways of going about fixing this. But yeah, let's not go into it. Yeah? Cool. So similarly, if we don't make a stimulus that is 10 times stronger or X times stronger, but we just have the same stimulus, but we reproduce it over time, like in repeated time. This is similar to what the question was about. We assume that signals add up linearly in time. So what that means is that if we have a brief event here, we get this Bode response, right? So there's a lot of time here, brief Bode response. That's what this would normally look like, right? But what happens if we start putting extra stimuli right after that initial stimulus? What you get is all these Bode responses after that. Yeah. Now, what we end up with is something that we can say we sum them all together, right? So that's the end result. Again, like in the summing of the strength, we can now sum all of these responses as they happen in time. And that gives us this compound Bode response. So I'm going to say that if I know the shape of the Bode response to a very brief event, and I have a very long block, I also can predict the shape of the response to that long block. Because nothing changes, it is a linear thing. I can just sum all of these responses together. Yeah. And this is what I need, right? So that what I'm explaining here is not necessarily a characteristic of how the Bode response works. In principle, this is mostly a trick that we use, an assumption, to allow us to use specific analysis techniques. Yeah. Right. So what this means is, for a given event, we have a specific response with a shape that we can either estimate or use. So we're now going to just assume it. And if we use this assumption of a linear time invariant system, there's a standard way in which we can start to predict responses, predict both responses from for example, an experiment, right? So we're just going to say, I'm showing a face here. What is the response that I predict? Well, that's called convolution. Many of you that are in the AI business will know convolution from convolutional neural networks. This is the same operation, but it is happening in time. So what this means is that we have an activation, a neural activation here. We have a bold response that we get from this, and we have what this hemodynamic response function is. So what happens, how do we create a prediction from the combination of our neural response and a bold response that we assume? It's what we do is we convolve them. What happens is we run this one through, we at every point in time, we multiply them together, and we take the value at that delay as the value that happens here, right? So this is just to show you the operation that happens. You always have to like reverse the shape of that one as you go through. You see is that clear? That's the operation that you're performing. There's math involved, but I'm sort of keeping it out for now. Yeah. So this is by assuming this hemodynamic response function and saying that this is the time at which my neurons activate. By performing this convolution, I can predict the time course of the bold response that I expect to find. Those are the different steps here. Yeah. Now, that might have looked a bit complicated. It's not really that complicated because you just tell Python to np.convolve, and then it's done. There are a few ins and outs that you will see during the second practical, but that's about it. So to create a expectation, like an expectation for the shape of the bold response over the time course of your experiment, it's actually very simple. You need to know when stuff happened in your experiment, and then you perform this convolution with this hemodynamic response function shape. Yeah. And so for a single event, that's what you just saw. Yeah. You start out with your neural activity that you expect, and then you get this bold response, right? So this is an impulse and this is the response. But you know, in a given experiment, you might have a very complicated experiment and you expect all sorts of graded neural activation, for example. Well, that's easy. You just input that over time. You perform this convolution with this bold response, and you get like a slower, smoother version of that same thing. Now, important to realize is that this gives us a way, this shortcut of using the convolution, right? It leverages these assumptions of linearity to be able to easily create expected fMRI time courses by just combining what we think is neural activations, which we have control over because it's our experiment. We impose this on the brain. Now I'm showing you a face. Now I'm showing you a house. We're using that, and we're using the shape of the hemodynamic response function that we can assume. Yeah, or we can measure it separately, and I'll get into that later. But those are the two ingredients. Yeah. Any questions about that? This is how we create expectations of what the bold response that we are measuring in the brain, what it should look like. Yeah. So we're modeling responses. There is a mathematical model behind what we expect our responses to look like. These 11 o'clock lectures are great in terms of timing at the start. The timing halfway through is when you actually, your body wants to have lunch, right? Don't you have that too? It's annoying. Well, there's no free lunch, right? It's either getting up early or being hungry during the lecture. No questions? Clear? Yes? Awesome. So then, having assumed that these responses all sum linearly, and we are looking at it like a linear time invariant signal, right? So the hemodynamic response function doesn't change, the response to events doesn't change, and we can use convolution to create predicted responses. We end up with the standard way of analyzing data, which is called using a GLM analysis, which stands for General Linear Model. So the meat of this type of analysis is that I'm showing you a very simple experiment, right? At random moments in time, I'm showing a house or a face, right? This is actually the average American house according to Google Images. And this is the average American face, I think, according to Google Images. In this analysis, you have these events occurring in time, and we've just convolved them with a standard hemodynamic response function. That's how I created this time course of expected signals. It's important to realize here we have two different types of events. Yeah. So what this means is that we have two different types of predicted responses, of expected responses. So we have this blue one, right? So all of these blue things, they reflect the signal that I expect to see in response to me showing a face. And the green one would be expected for a house. So we have, we can compare the brain responses to each of these separate responses and thereby adjudicate, is a voxel's response, is that due to the presentation of houses or to the presentation of, what was it? Faces. Yeah. Now, it's important to realize here that we are very explicitly creating predicted time courses. Right? We are assuming the HRF shape and then creating what we might call, or in many cases in different contexts, these are called explanatory variables, predictors, or regressors. Yeah. So all of those words reflect the idea that we use these assumptions to create a time course, a model time course that we predict. And now performing this GLM analysis then allows us to answer this question: for a given voxel, does the response of this voxel resemble the blue or the green pattern over time better? And is this voxel responding to a house or face?
Does this response also matter on which voxel you pick? So say you have something that's about the memory, and you can have certain memories to a certain type of house. Would that give a different?
Yes.
Signal?
It's important to realize, though, that that is not a change. So you might expect that this type of machinery method or mechanism works differently in a different type of in different part of the brain, for example. Indeed, there could be like repetitions of a given face somewhere, right? Where or some faces are different. That's cool. You can do this experiment. The important insight here, though, is that what we are doing is we're modeling things, right? And so if I've already run this experiment, what I can do is I can change my model where the the sort of modeled neural activity, right? The predicted neural activity is not the same for all of the presentations of all these faces. It's not an identical response, but the response depends on X, where X could be memory, or X could be male or female, or X could be average or idiosyncratic or whatever, right? And so what we can do is run a given experiment. And if our experiment has these differences in it, then we can tailor our analysis, our modeling, to be able to find these differences. But then again, this is we'll get to this in later lectures, like how you use this statistically and that sort of thing. But indeed, that is the logic, right? By modeling responses, we give ourselves a playground, and we can use that in analyses. Okay? Now, how this goes into statistics and what have you, we'll get to in later lectures, right? This is just for the concepts. Yeah?
Just want to clarify something from what you said. So if I'm understanding you correctly, you're saying the assumption is always that you get the same signal, but at different points of time, and at what point of time you're coordinating which part of which pixels, and that's when you say corresponding to different objects that you're showing, maybe.
Yeah. So to be clear, right? Let's say because I showed you these blobs, right? I showed you a house blob and a face blob in the brain, right? What this means is, this is the result of this analysis that I'm showing you. So what this means is that if I go into one of these orange blobs, what you will see is that the response, I forget, orange was houses, right? So orange is green. And what that means is that if I look into a blob like that, and I look at the time course of the response, it will look like the green time course here, right? If I look in the blue blob, which was blue is blue. I know there was a reason for this somewhere. Then you see this, like if you look at the time course of the voxels in that region, you will see it resemble the blue time course, right? And so what the GLM analysis allows us to do is say, this is a house voxel, this is a face voxel, because it quantifies the resemblance of a voxel's time course to each of these predicted time courses. Yeah.
How different are the models and responses in comparison to the actual published models?
Ah, this depends on when you showed the houses and when you showed the faces. So this is it's a matter of experimental design in many cases. Yeah. But this is important, right? Because you can imagine doing an experiment where it becomes impossible to differentiate the responses to a face and a house. For example, if I always do house face, 20 seconds, nothing, house face, right? How am I going to tell them apart? Because the bold responses are going to completely overlap, right? And so what this means is that when I do an experiment, and we'll get into this, there's like an experimental design lecture later on, right? But you see the intricacies here, that if you manage to make these two predicted time courses, right, design your experiment such that these predicted time courses look maximally dissimilar in some metric, then you have a powerful experiment, right? Because then it becomes easy to say for a given measured signal, it resembles blue or red, or sorry, a green. Geez, I'm like language colorblind.
Also a form of validation.
Yes, and so what it means is that you can actually simulate experimental designs, like in what order and at what timing am I going to show houses and faces to be able to maximally get out a difference that I'm interested in. Yeah, and so here you see that this is very model based, right? Yeah, and it gives you a lot of power, right? You can actually, really think deeply about how you want the experiment to work, and then present exactly that experiment. But there's a lot of thought that goes into this, and we'll get into that in later lectures.
But this would limit us in terms of we wouldn't know, or it'd be harder for us to know if the response is because of the house or some certain features in the image of the house.
Yeah, this is why I'm sort of showing you the average here, because I was afraid that you might think that it is because of specificity of a specific stimulus. It might be, right? And if you're interested in, is there a difference between like flats, like high-rise and low-rise buildings, or whether it's in a rich neighborhood or a poor neighborhood, you can do that experiment, right? And then you would just have, blue would be rich houses and green would be poor houses or something like that, right? It's all a matter of what you want to test for in your experiment. Yeah. Is that clear? Awesome. And so the GLM analysis is all about setting things up such that you can say, this is a house voxel, this is a face voxel. Geez, I get confused with this. Yeah, so, and so the rest of the lecture is just me trying to explain the general sort of geometric principle of what this GLM analysis is. And so any of you that have done like a deep learning course or like a machine learning or data analysis course in a computer science department, that sort of thing, are going to think this is completely trivial. So I'm just taking this very, very slowly because it's very important that you realize the ins and outs here, right? And there are certain caveats that come into play later on that you don't generally think of when you have this sort of AI type background. But if you have that background, then it's going to be easier for you to just write a line of Python code that performs this analysis. But we're going to go slowly for the concepts. So this arrow here is just there for nothing. No, that's not true. There's a step in the analysis where we take these regressors, right? We call them that. I might call them explanatory variables. I might call them, add another word, I always forget. I'll call them regressors from now on. So these are expectations of what the time shape of a signal will look like. We call those regressors. And I'm going to refer to them here as X1 and X2. Yeah, so I'm going to try to put in a bit of math lingo, but not too much. And so my task here is then to take a measurement, right? So this is a single voxel time course of data of bold signals that I'm recording from some portion of the brain, right? And I'm calling that Y, and this is a subscript 1 because it's the first voxel that we're looking at. So you have to remember here that this one and this one are different things. X1 is the first regressor, Y1 is the first voxel. I'll go into that in a bit. So to be very clear, right, our goal in this analysis is to try to explain Y as a function of some linear combination of X's. Yeah. A linear combination, I'll get into that in a bit, but that also has to do with this LTI assumption, right? It has to be a linear combination. So that's our goal. We have regressors or explanatory variables, and our goal is to predict this time course, to fit this time course. Yeah. Now, there is an equation that pops up in the wrong moment here. So, yeah. So just as a to be clear, right? Y is on the left-hand side here, on the right-hand side there, right? So I'm trying to explain the signal Y. Yeah? And I'm trying to do that as a combination of X1 and X2, the blue and the green one. Yeah. So that should give me a nice fit to explain everything that happens in the red. I'm needing to add a C here because I don't know where this line is, right? Like I said, the bolt response number, right? It's an arbitrary number. It can be at 10,000, it can be at 0, it can be at minus 2,000, although generally isn't negative, but you get the point, right? It's an arbitrary value. So I also need to be able to place this curve anywhere. So I can add some number. And of course, then there's noise. So my model of what produces this response is some combination of the interesting regressors over time and what we call an intercept. So that's putting it at a different level and a measure of noise. Yeah. Now, each of these A, B, and C are regressors. So even the intercept, which is like a flat line, ones everywhere, it's not interesting, but it can be scaled by a factor, right? That's that C. And so these we call beta weights. They are scaling factors, meaning that we get a number and it tells us how we should scale the blue line, the green line, and the flat line to best fit this bolt response. So this is the second step of that LTI, what that LTI system assumption allows us to do. The first one is we can convolve with the hemodynamic response function that we assume, right? The second is we can now think that this is produced by a linear combination of these interesting things. And again, if we have changing, if we change the model, what we do is we change this part of this whole approach. We can always reapply that to that. So we have a lot of leeway in shaping our analysis because we can model things differently. Our inferences depend on that. Yeah. Now, in the end, of course, we get these beta weights, and what they do is they relate to that question of interest. Is this voxel a house voxel or is it a face voxel, right? Because if I get a very large AI, right, so that is the blue time course needs to be scaled by a large number in order to fit this time course. And for example, the B is low, so that the green time course needs to be scaled by a low number. Yeah, I have a large beta weight for the blue, have a small beta weight for the green. So these beta weights, they relate to whether a voxel is a house or face. This pattern would reverse if it's not a house voxel, but a face voxel, for example. Yeah. And so I just said this, but let me try to go through this in a bit more visual way. Right, so these beta weights, what they come out of the analysis. I'll talk a bit about how you fit this sort of thing, but they come out of the analysis and they're the values that you need to multiply your EVs with, right, so scaling factors, in order to best explain the data. So we have a measured bolt response. We want to explain it. What values do we need to give these beta variables? And they are going to go into the next stage of the analysis because they allow us to say whether something is a house or a face value, right? And so our explanation of the data is what we call Y hat. So you'll see Y hat appear over the course a couple of times. Y hat is our best fitting explanation of the data. That is, we find a beta weight for each of those things, and we can add them together with the regressors, and they are the time course that we model. That's the resulting time course that we find. So just to put it in a simple picture here, Y1 was our experimental data, right? The red line was the voxel response that we recorded, and You see here, the black dashed line is our best fitting prediction. So this black line is composed of these expectations that we had beforehand, multiplied by fitted beta weights. I'll go into how we fit those beta weights later, but this is how we analyze fMRI data. This is how we compare our model to the data, right? It goes through this eventual predicted response of y hat. Now, what does this work like? I made a little movie that I'm terribly proud of, so please don't laugh. So we can scale these beta weights, right? Here you see the values of the different beta weights. You see that this dashed line is changed as a result of changing these beta weights. You see the beta times x, you see them change here, and you see the result of y hat changing. And at some point, they're at their optimal values, right? So it's sort of, I'm pretending that I'm fitting these responses, right? And now you see that I've ended up with a best fitting response. Is that clear? I'm going to show it again because I'm so proud of it. Right, so you see that we're tailoring the beta weights, right? And what that does is it changes the scalings of these different expected values for each of the different types of events. They're added together, and that allows us to tailor this predicted y hat signal. Okay? And our goal is to make it look as much as possible as these, as the red line. That's the goal. Our end result is that, yeah?
I still didn't understand where the X2's like expected value signals are coming from.
Oh yeah, so they're there because I showed the face at time point 24, for example. I used the bold response to create a predicted signal for a house, right? And faces I showed at time point 36, for example. And so that's when that regressor gets its HRF shape, right? And so I have a model in my head, right, of what a face response should look like. Yeah, it's triggered at time point 24 and not at 36, right? It's time, and I have a bunch of these time points, and together this gives me like the blue prediction. Yeah, and similarly for the green, right? There's just different time points, same procedure otherwise. And so what this analysis does is it tries to establish how much of the green and the blue I need to add together to produce the red, namely the data that I have. Does that clarify things?
Yeah, so it's like for this voxel, you get those scalars beta 1 and 2, you have them saying like that's the ratio of the activation explained by
Exactly. Yeah, and you see that in the corner here. Here, beta 1 is 3 times the value of beta 2. So this is 3 times as much a house voxel as it is a face voxel. That statement we could argue about because you go into statistics and all that, and we'll go into that later on. But that would be the way of quantifying things, right? This is fitting those beta weights. Okay. Any questions about this?
So which voxels do you use to make this prediction? Next one and next two.
Excellent question. None.
Oh.
So this is a good question because there is a conceptual Berlin Wall between the part of the analysis that uses data, right, and the part of the analysis that produces the models. The models only use what you know about the experiment, when shit happened, right, and the shape of the hemodynamic response function. That's what you use to create these predictors. It's important also to realize then that this is identical for all voxels throughout the brain. They are independent from the voxels. You can do this on a simulation. You can do an analysis on a simulation, no problem. Like I did show the averaging, the epoch. And so, you have a lot of leeway in producing a specific model, right? It's just sort of, you think like, oh, but I think there's a difference between male and female faces, or rich and poor houses. That changes the model side of things. Okay? And so then you apply it to the brain, where you fit these beta weights, and you actually do exactly the same analysis for all voxels throughout the brain. Yes. You really need to conceptually separate these two different phases of the analysis. The one is applied to the data in parallel across the entire brain. You don't even care, right? The outcome will just show itself in terms of these beta weights that are going to be the result of that analysis. And then on the other side, you have all the hard work of modeling this thing, right? And it depends on your shape of choice for the hemodynamic response function. It depends on whether you think that all faces should be considered equal, right? All of those things, but they are a separate arena, if you will. Yeah. Is that clear? Because it's a very important conceptual point. Yeah. The data do not figure into this, the phrasing of this analysis. Only when we fit the beta weights do we get an attachment to the data, if you will. Yeah. Hmm?
So the modeling part, we're going to do it at a voxel level, I think.
But it kind of depends on what you call the modeling part. Constructing the regressors, for example, is done before. Yeah. Only when you want to compare your expectations, right? So these blue, what? Geez. Oh, I forget where I am. Oh, the beautiful movie again. So these standardized ones, right? The ones that I just created, they're the same height. They come from my experiment, right? And they just exist before I do any data analysis. And then this procedure of finding the beta weights, they are the actual model fitting. They are what you could call the actual analysis. Yeah. The first part is modeling. That's just imaginary.
I'm talking about the blue and the green lines that we decided before.
Yeah.
That would be depending on like is it how we model, and that would change probably from regions or voxels.
Yes, yes. So, but that shows itself in the outcome of the analysis, right? And of course, if you are modeling a certain difference, then that allows you to find the difference in the brain. And if you don't model that difference, you are blind to that difference in the brain. Yeah? So you need to be able to first come up with a potential difference in order to then find it, right? I could also have said, and just to take this to the extreme, right? I could have said, yeah, well, I'm just going to pretend that I have only a single type of response, and it's going to be 50-50 house and face, right? So I'm just going to take my X1 and X2 and just add them together, and I won't be able to do any analysis anymore, right? I just get to see whether something is a house face voxel, right? And I won't be able to tell the difference anymore. So I need to have these separate regressors in order to be able to find the difference. Yeah. Cool. And I think I'll go through this a bit. It becomes clearer as we start piling on repercussions, trust me. Yeah. Now, so this is what that looks like. This is our explained signal, a Y hat. It's very important because it allows you to see how, for a given voxel, the response of that voxel aligns with your model, right? And so we talk in this case about variance a lot. We want to explain variance in the data maximally. We want to explain as much variance in the signal as possible. So, and the better our model is, the higher this ratio of explained variance becomes. And so, just to highlight that, right? And in this case, there wasn't so much noise. Noise will add variance that I cannot explain. In this case, there was a very good model fit, right? You see, there's hardly any discrepancy between Y hat and Y. Y still being red and Y hat being the dashed line. And so this is the noise distribution over time. It's very important to sometimes inspect this, right? Because it might be the case that you have noise that just has this really weird wobble at some point during your experiment. So it's the structure of what we call the residuals, which is this, the noise, can be very, very informative. We'll get to that later on. Yeah. This is just to show you what a very good model fit would look like. And then just let me replace that with a bad model fit, in which case you have these periods inside your data that you are not explaining with this best fitting model. And this means that there is more variance that you are not explaining. Yeah? Your unexplained variance goes up. And you see that this distribution becomes wider, and we don't want that. So the idea of most fMRI analysis depends on this principle. You are on a quest as an analyzer of data. You are on a quest to explain variance. So you have a signal that goes up and down, and the better you can explain the ups and downs of that signal, the better you are as an analyzer, the better your data are. And so your quest means that your tools are to create better models, right? That is your quest, that's your tool, modeling. Your enemy is unexplained variance. Okay? That's the part of the signal that you are desperately eager to kill. Yeah? And I'll go into this over and over. But that's the idea. Yeah. Any questions about that?
What is a good and a bad variance? What is the sign?
Ah, yes, statistics. So there are multiple ways in which we can do this. So one is inferential statistics, which is like more in the sort of localization of brain activation approach where, and I'll go into this in a bit if I have time, but you can say, oh, how much of the signal am I not explaining? How much am I explaining? The ratio, depending on, a couple of factors go in. But you say you create a t-statistic and that gives you a p-value for a given voxel, right? So that's when statistics come in. Another approach, which is gaining more and more sort of use, is to cross-validate. So you fit, you get beta weights, you get a separate run with a different set of timings, and then you say, how well am I predicting this without having fit the beta weights? And so that allows you to see. Then the measure that you use is cross-validated explained variance, out of set explained variance. I'll get into all of that later on, right? So that's towards the latter portions of the course that sort of thing becomes more important. For now, what you need to know is our modeling approach, right? Our explicit definition of now things happen. I'm using this HRF. I have an explicit prediction of what the signal should look like. These are my regressors and they go into my analysis. That is the logic that we use here. And our goal is to explain as much of the signal as possible. Yeah. As much as possible is quantified by means of variance. Clear? Awesome. Now, of course, we do this thing not just for single voxels, right? That would take a lot of time. Yeah, because we have in a given measurement, like 100, 150,000, 300,000 voxels. And so iterating this over all voxels would take a lot of time. But it turns out that computer people are really smart about this. We can actually perform this analysis in parallel, right? So computer processors and GPUs and all these types of things, they are made to perform similar or identical calculations on oodles of data very, very fast. And this means that performing an analysis on a single voxel isn't even that much slower than performing it on thousands of voxels. So we're in luck. So what does this look like if we take into account this sort of shape of the data? We add a dimension now, right? So we've just been looking only at single voxels, and now we're going to add multiple voxels. We can still do it in the same way, but I just want to highlight what the data then looks like. So again, Y here is the data. X is the set of regressors that we're going to use to explain the data all throughout the brain. The beta weights are the things that we fit. And of course, we have noise, which results in an error term, right? The stuff that we cannot explain. Again, E stands for enemy here, right? So what does this look like, right? Just to look at the shape of the matrices that go into this type of computation. And you'll see this as you go through the second practical, how this works if you implement it in Python. Yeah. And so in this case, in an analysis like this, we could not care less about where a voxel is. They are interchangeable. Yeah? They are just voxels, each has a time course. And so what we do is we flatten out the entire brain. Yeah. We don't care about things. We have a separate routine that tells us where they were in the brain in the first place, but for the analysis, it does not matter where the voxels are. So we flatten it out. And that means what goes into the analysis is a two-dimensional matrix, right? Not a four-dimensional matrix. So if you were scared of having to use 4 dimensions in NumPy for your analysis, you're in luck, right? We can just flatten it out. We have voxels by time. That goes into the data analysis. That's our Y. That means that is our bold signal that we are trying to explain. It's the data that you collect is on the left-hand side of the equal sign. Yeah. Now, what do we have here? We have our regressors at X. Yeah, those are where X1, X2, and the intercept. Yeah, the intercept being just ones allowed us to scale things up and down. So this is our model. We call this a design matrix. So this is the same across all voxels. The analysis uses only a single design matrix. What that means is that it has time as one dimension and regressors, only regressors as another dimension. Yeah. This structure is clear, right? And if not, you can ask. So important concept here, design matrix, and we always refer to it as X. Right? So this is these regressors that we've been talking about. That's what's in X. Now, what we want to find here is betas. Yeah? And what is betas in the end? What comes out of this analysis? Well, you get a beta weight for every regressor, right? And you get a beta weight for every voxel. Because you're running this analysis in parallel across all voxels, and you are going to get a beta weight for every regressor. So that means that the beta outcome matrix of this analysis, if we run it over the entire brain, is composed of voxels in one direction and parameter weights, beta weights, in the other direction. Yeah. And of course, taking these together gives us that y hat, right? If you go back to the equation on the previous slide, just dot producting this, and we'll go into all of that later on. But these together give you the predicted signal over time that you can compare with the actual signal. And then the difference is the residuals, the noise that you, this part of the signal that you do not explain with this analysis. Now, this is again shaped exactly like the incoming signal, right? Because the combination of the design matrix and the beta weights just sets up a portion of the signal that you can subtract from the original data to give you the unexplained variance. Yeah? So we have a time course for every voxel in the brain of shit that we hate, right? The stuff that we couldn't explain. It's so frustrating. You see it? Right? This is all the stuff that you have no handle on. It's when your subject was coughing in the scanner and you didn't hear because the machine is so loud, right? Signals going up and down throughout the brain and you didn't know. So it's going to destroy your analysis because all of that variance is going to end up in stuff that you cannot explain. So your goal is to explain this stuff. We'll go into specifics later on. So quickly, I think I'm running out of time, so I'll go into a bit of this, how you implement it, right, in Python, because we're going to put the second practical online in a couple of days, and you'll need this next couple of slides, and then we'll pick it up in a week from now to go into specifics further. And so standard GLM analysis for fMRI looks like this. So this is like a matrix equation where Y is a matrix, X is a matrix, beta is a matrix, and E is a matrix, right? And so the error is just normally distributed, we might propose. And if you assume that this is all the case, right? Oh, sorry, yeah. So our goal is to make sure that we predict most of the signal, right? And what we're going to minimize is the squared difference between our best fitting signal and the data. And so this these errors that we have, what we're going to do is we are going to square them and add them all together, and that's going to be how bad we feel. Now, it turns out that it's just very, very simple to do this in a computer. If you take this equation and you implement it into standard linear algebra, then it becomes sort of this equation. If your best estimate of the beta weights is just some operation that you perform on the design matrix multiplied by Y. And you don't have to care about this necessarily. I'm not going to quiz you on the specifics of this equation. You just need to know that it's easy to do. Yeah? Well, that helps. Now, one final thing about this idea of statistics and how we want to explain variance quickly. We want to decrease this error, right? The error of enemy, the E of enemy. That error we want to take down. And so generally what we do in inferential statistics, we are interested in blobology, in localizing brain activation. We want to create a t-statistic. This is the equation for the t-statistic. It's just our beta weights that we fit divided by the standard error. And that standard error, you see, it depends on the errors. So this is that squared errors. The errors here are squared. This is Y hat. This is Y. We take the difference, we square them, and we sum them, right? So how much of the variance can we not explain? And then we divide that by the degrees of freedom. We'll go into all of that. But the idea here is that in the end, geez, Louise, what happened? In the end, we can look at this and immediately see where we want to be, right? We are modeling these beta waves, right? They are a result from our model. And we want this standard error to be as small as possible. Just look at this fraction, right? You want this part to go down because it means that your t-statistics go up. Right? The higher your t-statistic, the more certain you can be that something, a difference, for example, that you were looking at is there. So this is just a very, very simple rendition of really the idea that we are trying to explain as much variance as possible. I think we all have to run now. But seriously, yeah, I hope this highlights the importance of getting a good model fit, because it increases this numerator, right? And explaining as much of the variance as possible, because that decreases that denominator of this t-value fraction. Yeah. The practical will go into detail more, give you an idea of how we implement this sort of thing in just a couple of lines of code. And so the next lecture is on Wednesday is going to be MR physics. So we're going to take a step back and talk about how you create images in any way from these radio waves and magnets. And then on Friday next week, we're going to go into statistics of localization and some more of these types of concepts. Okay, have a great weekend. Lunch. Yay.
Yeah.